So welcome back. With the innate immune defenses that we talked about those that are um, those that we do not have to develop on our own. They are present in us at birth, and they are very non-specific in nature. We'll now move into the adaptive immune defenses and exactly how those work, and we'll see that now they are going to be very specific, and we have to develop them as we encounter new foreign organisms and pathogens. So these are going to be our third line of defense. So we had first line of defense was innate with our barrier defenses. We had second line with cells and chemicals, which were also innate. And now we have our third line of defense with our adaptive immune defenses. And these are going to be very specific and target specific types of antigens and pathogens. So we can classify our adaptive immune defense into two broad categories. We have humoral immunity, which is sometimes called antibody mediated, and then we have our cellular immunity, which is sometimes referred to as cell mediated immunity. Humoral immunity is going to involve our B lymphocytes, and they will ultimately differentiate into something we'll call a plasma cell, and then that plasma cell will secrete antibodies. And then we'll have our cellular immunity, which is going to involve our T lymphocytes, and we'll see that there are different types of T lymphocytes and how they will interact with our B cells to induce an immune reaction. So these cells, B cells and T cells, they exist in humoral immunity and cell immunity, but they interact with each other. Especially in cellular immunity, you'll see the use of B cells in both instances. So let's define some terms first before we get into it. So an antigen is going to be some type of molecule or some type of surface marker that our immune system will recognize as either being foreign or non-self, and then self. So foreign and non-self mean the same thing. They just mean something that is not native to our body. And then a self-antigen is a marker on our cell which is native to us. So the word antigen, when you break it down, it means anti. The anti part means antibody, and then the gen means generator. So these are molecules that generate the formation of antibodies. Now, on the surface of an antigen, there is a very specific point that is called an antigenic determinant, and this is also referred to as the epitope. It's a smaller component of an antigen, and it is actually what our immune system recognizes and response to. So the whole molecule generally is referred to as an antigen, and then the specific part on that antigen is going to be the epitope. Okay. Now, we have these antigens on our cells, and they will be presented to our other cells on a protein called a major histocompatibility complex protein, sometimes called an MHC. So the major histocompatibility complex, it's a region, it's a gene, essentially. It's a region of our DNA that codes for these MHC proteins. And we'll see that these proteins will present these antigens on a cell called an antigen-presenting cell. So the naming scheme really makes sense. These antigen-presenting cells are going to present these antigens on one of these major histocompatibility complex proteins. And we'll see that there are different types of major histocompatibility complex proteins. Now, here you can see a bacterium that is being phagocytized and brought into to a macrophage or a phagocytic cell. It fuses with the lysosome, and that breaks it down. And then the little fragments of that are going to be placed on one of these MHC proteins and then presented on this cell. So this type of cell here, since it is presenting an antigen on its surface, this is going to be an antigen-presenting cell, something we'll call an APC, which stands for antigen-presenting cell. Now, when we talk about our adaptive immune system, like I mentioned a second ago, we're going to have our lymphocytes, which is a type of leukocyte, remember. Lymphocytes is a subset of leukocyte. Leukocyte is just the overall term for a white blood cell. These types of lymphocytes are going to be our T cells, which mature in the thymus, and then the B cells, which mature in the bone marrow, and they both originate in the bone marrow, if you remember. T cells will be involved in our cell-mediated immunity, and our B cells be involved in our humoral immunity. So this is a picture that is showing just an overview of what's going to be going on in humoral immunity and cellular immunity, and then there are helper cells which kind of interact between and are fixated between humoral and cell-mediated immunity. So note, with humoral, you use the use of B cells and plasma cells with antibodies, and then our cell-mediated or cellular immunity, we're using T cells here that will actually specifically target our foreign or infected cells. So this idea of having our immune cells target foreign antigens kind of gives us the impression that we have to somehow be able to not target self-antigens. So we don't want to target cells and antigens in our body that are that are native to us. So our cells display this idea of self-tolerance, in which we don't have our cells respond to our own tissues, and we actively get rid of and destroy cells that ultimately would respond to our own tissue. So with T cells, there's positive and negative selection, in which you positively select ones that will target foreign tissues and foreign antigens, which is good, and then negatively select, you get rid of the ones that would target self cells. So it's just this idea, you can read about it in your textbook, of keeping cells that we want that will target foreign antigens and getting rid of those cells that will ultimately respond to our own cells and tissues. And then immunocompetence is referring to the idea that our lymphocyte is capable of binding to an antigen. That doesn't mean that it has yet, but it has the ability to, and there will be receptors on its surface that can actually bind to these antigens and proceed with the immune reaction. So the way that we get all of our, if you think back to how many different types of bacteria and viruses and parasites and different foreign antigens that we could possibly encounter, there are millions and millions of different things that our body could encounter that would be considered foreign, okay? So we have our genetic information stored in our DNA, and this genetic information in DNA will go to RNA, and then this RNA will ultimately make proteins, and our surface receptors are different types of proteins, right? So the way we get all of this combination of different types of protein receptors that will respond to different antigens is we do something called somatic recombination. And what it is we take a section of our gene that codes for these proteins. And if you remember, in DNA you have A, T, C, and G, so adenine, thymine, guanine, and cytosine. And then in RNA you have uracil, you have uracil, adenine, guanine, and cytosine. And what you do with these is you shuffle up and you shuffle around these different nitrogenous bases to get a different gene segment so you can code for different proteins. And this leads to this idea of us being able to have a wide variety of receptors to respond to a wide variety of different types of antigens. So we have on the order of 10 to the 11th different receptors that can act and target different antigens because of these different receptors that have been produced by somatic recombination. So we can get more than 1 billion different types of receptors just by intentionally shuffling specific sections of a gene. Okay, so we'll start first by talking about something called an antigen presenting cell. So an antigen presenting cell is doing exactly what it sounds like in its name. It is presenting antigens to the immune system. And we have 3 main types of antigen presenting cells. We have macrophages, which we've talked about a little bit. We have B cells or B lymphocytes, and then we have something called a dendritic cell. Okay, so all 3 of these will undergo this process of bringing in an antigen, processing it and breaking it down, and then presenting it on its cell surface via an MHC, that major histocompatibility complex protein or molecule. Now, there are 2 types of MHC complexes, as I alluded to. There is a MHC class 1, and there's an MHC class 2. MHC class 2s are going to be on the surface of our antigen presenting cells, and they are what will contain the the small fragment of the antigen that will be presented. So when you think of antigen presenting cells, these macrophages, B cells, and dendritic cells, think of them presenting the processed antigen on an MHC2 complex, and then that'll be presented to our immune system, to these T cells that will then act and respond to it. So here you can see our dendritic cell, which these dendritic cells are found in the skin. They're specifically found in the stratum spinosum of our epidermis. We also have our macrophages, and then we have our B cells, which we've talked about. All three of these are going to be antigen-presenting cells, and they will present their antigen on a MHC class II molecule. Now, these B cells that we talked about, these are going to be involved with our humoral immunity, or our humoral immune response. Now, we have antigens just circulating in our blood. Okay, they're circulating if they've entered through a break in the skin or somehow otherwise entered into our bodies. And what they'll do is they'll encounter a B cell and they'll bind to the B cell's receptors. And so a B cell receptor is just an antibody. And we'll look at what antibodies are, their structure, and how there are different classes of them. But what they do is essentially after a B cell has to become activated, but once it binds to the receptor and it is activated, the B cell will divide into two different types of cells. One is a memory cell, which will be basically the same thing as that B cell itself. And what it serves to do is it stays around in circulation for a possible future encounter with that same antigen, so that we already have memory cells ready to respond. And so we have these memory cells that recognize the same, and it has to be the same antigen as what encountered the initial B cell, but they are present if we happen to encounter a antigen that is the exact same somewhere down the line. The second type of cell that they can differentiate into is called a plasma cell. Now, a plasma cell are going to be the antibody factories, if you will. They secrete lots and lots of antibodies every second. So on the order of 2,000 antibodies every second can be released from a plasma cell. And these plasma cells are derived from B cells. Okay, so don't say that B cells are the ones that secrete the antibodies. Say specifically that the plasma cells are those that secrete antibodies. So here's just a basic picture showing how our B cells can differentiate. They can go from just the normal B cell and then ultimately transition either into a memory B cell or a antibody secreting plasma cell. Now, this memory that we talked about, we have different responses, different levels of immunity, if you will, depending on whether we're encountering something the first time or if we're encountering something for the second time. So our primary immune response is going to be the very first time we ever encounter some antigen, okay, some foreign thing. That's the very first time we encounter it. This is a primary immune response. So you can imagine if we've never experienced that antigen before, if we've never encountered it, we're not going to have very high antibody levels because we've never seen it before. So that's exactly what we see. The antibody levels are generally low. But when we see our secondary immune reaction or immune response, we now see an increase in antibody levels because now we already have memory cells, and those memory cells upon the second encounter can differentiate into plasma cells, and then those plasma cells now secrete lots of antibodies. So you get a greater antibody level, and you have less of a time delay between the reaction and the immune response because now you've already encountered it once before, so you're essentially primed to be able to recognize it upon a secondary exposure. So what you can see here is this graph is a really important graph. You can see how there is an initial exposure down here. You have your response, the primary response. Then your antibody levels will drop off. Now, the second time you encounter it, your antibody levels are already higher than they were the very first time before you were initially exposed. The second time you get exposed, now you see a very sharp and drastic increase for our secondary immune response. And now note the concentration of antibodies. Now it is much, much greater than it was during our primary immune response. And this is because we've already encountered it for the first time. We already have our memory cells and the antibodies from our first round of plasma cells that secreted these antibodies. So we're essentially primed and we're ready. And so we have a more drastic and greater secondary immune response as compared to our first immune response. Okay, so we have different types of immunity in terms of how we get these antibodies. So here, if we go back, we're talking about concentration of antibodies and how many there are. Well, how do we get those? So we can get them in different ways. We can get them actively, or we can get them passively. Actively means that we actually have to produce the antibodies after an immune encounter, an immune response. Passive immunity is referring to the idea that we are directly given, or we directly receive the antibodies, and we don't have to actually have an immune response to make those. Now, with active immunity and with passive immunity, there are two variations. You can have natural and artificial. So you can have natural active and artificial active. You can also have natural passive and artificial passive. Okay. Natural for both passive and active mean we get it naturally. Okay, so we're not getting it artificially through some outside source like at a hospital or a clinic or something like that. It's completely natural. So for natural active, it's just a normal infection. You get infected with the virus, with the bacteria, you're immune. You have an immune response, whether it's probably a primary immune response, and you get antibody production. So you're actually making and producing the antibodies. Artificial is something like a vaccine. So you're given an attenuated or an inactive virus. Your immune system still recognizes the antigens on that, and they actively produce antibodies. But the stimulus for them to make it is now artificial because it was given to you from an outside source via a vaccine. Okay, so hopefully that makes sense. With passive, we said they're just directly administered, the antibodies are. And so for natural passive, this is going to occur commonly between a mother and a fetus and her fetus. So antibodies are able to pass through the placenta to the mother. And so this is a way in which you can have natural, because this is a natural process, but passive in terms of the fetus receiving the antibodies. The fetus did not have to actively make the antibodies. They were directly administered to it. There can also be antibodies passed in the breast milk to a newborn baby as well. Now, for artificial passive, this is going to be you are given directly antibodies that is in an artificial sense. So you get antibody therapy at a hospital or clinic. So these antibodies are directly given to you. You did not have to make them. Okay. So hopefully, whenever you study these active and passive, think about what active and passive mean. Active, you're having to actively make the antibodies. Passive, you're just passively receiving them. And then think about natural and artificial. Is this a natural response or a natural means of receiving or making antibodies, or is it an artificial means of either making them or receiving them? So here you can see our variations. So natural active is going to be an infection. Artificial active will be a vaccine, so with something maybe like the COVID vaccine. For natural passive, passing of antibodies between the mother and her baby. And then artificial passive, you just get antibody therapy. So antibodies are directly administered into your bloodstream. So let's talk about these antibodies a little bit more and exactly what their structure is. So you generally have two main chains on an antibody. You have a heavy chain and a light chain. And then within these, you have something called a variable region and a constant region. So you can think that the constant region is going to be essentially constant between all antibodies. And then the variable region, and so these antibodies are proteins. Okay, so the variable region is going to have different amino acids in a different sequence that makes them different from another type of antibody. So it gives them specificity, if you will. These antibodies can be freely circulating in your blood, just circulating around, and when they encounter a pathogen, they bind to it. They are also the receptors on our B cells. So when a B cell binds a foreign antigen or pathogen that is floating around in the blood, in our bloodstream, what it will bind to is a B cell receptor, which is an antibody. And these antibodies are also sometimes referred to as immunoglobulins. Okay. And so antibodies also have this ability to switch from one type. So we'll talk about how there are there's M, A, D, G, and E antibodies. They can switch between different types, but the specificity of what antigen they respond to and are specific for, that doesn't change. Whether it's A or M or D or G or E, the specificity remains the same, but it's the class of antibody that can actually change. So when we look here, what you can see is a small light chain on this outside portion. You can then see a larger heavy chain along the middle. And then you can see a variable region, which is this more superior region. And then all of this down below, this is essentially constant between all antibodies. So this is where the antigen would bind to on this antigen binding site along the variable region. And then this is a more accurate 3D rendering of what a actual antibody would look like. This is how antibodies are generally drawn in this kind of Y shape. And so I'd be able to look at an antibody and pick out, which regions are variable and constant, where the light and heavy chains are, and where an antigen would actually bind. So here are different antibody classes, and so these classes of antibodies will be secreted during different scenarios. Okay, so antibody, and there's some, I said they're called immunoglobulins, so that's what the IG refers to: IgM, IgG, IgA, IgE, and IgD. These are secreted and released in different situations. So during our initial immune response, that very first immune response, the primary antibody that is released is IgM. Okay, and it really has, it's a series of 5 antibodies kind of stuck together, and each antibody has 2 binding sites. So it is going to have 10 binding sites total, and these are what can actively be secreted from our B cells and our plasma cells. Now, with our secondary immune response, so the second time we encounter it, the primary antibody now is going to be IgG. And these are the most common antibody in our body, and these are actually what cross the placenta and can travel from the mother to the fetus. IgA are going to be found in different secretions, so like mucus, saliva, tears, and then colostrum. Colostrum is essentially just the first amount of breast milk that is produced after pregnancy. IgE is going to be associated with allergies and anaphylaxis, and it's going to be generally very low. It's not a very common antibody, but these can bind to cells called mast cells and mast cells and result in histamine release. So you've all heard of an antihistamine. What an antihistamine does is it binds to the receptors on mast cells where these IgE antibodies would bind to. And so when you block the receptor, you prevent IgE from working, and IgE causes histamine release. So an antihistamine essentially prevents the release of histamine via the blockage of receptors for IgE. And then IgD, the last one here, are going to just be the B cell receptors that we find on our B lymphocytes. So what you can see here are our different types of immunoglobulins and their relative percentages in the body. Note the IgM, how it's really 5 stuck together. It's really 5 IgGs stuck together. And what you have is 2 binding sites on each antibody, so you have a total of 10. And then you have most of these, and they all look relatively similar, and so you can note the light chain and the heavy chain, where the constant region would be and where the variable portion would be as well. You can see their percentages down here, as well as what they are secreted in response to. So this bottom portion down here, function, is basically the same thing we have written right here under our antibody classes. So our antibodies can perform various functions and can interact together to result in different products. Okay, so we can have antigens and antibodies interact to form something called an antigen-antibody complex. And with this complex, we have different subtypes, essentially. So we have neutralization. This occurs when antibodies travel to the pathogen, and where that pathogen will ultimately bind to our cell and cause damage, it's going to block those sites, and so the pathogen is unable to perform its function and exert its effects on our bodies. Agglutination is essentially when you have many antibodies kind of form these cross links and connections, and so you'll have one single antigen generally, and then you'll have multiple antibodies form all of these cross links together to essentially cause this clumping. Precipitation is similar to agglutination, but what happens is you have these cross links form that are so large that it actually forms an insoluble precipitate, is what it's called, that will settle out of solution. So it's similar to agglutination, but just a little bit larger. And then we have our complement fixation, and this has to do with the complement system that we talked about with our innate and nonspecific immunity, where you have antibodies bind to an antigen, and then this stimulates the complement system, ultimately leading to the lysis of a cell via the influx of water, resulting in its destruction. So here you can see some of our different things. You have neutralization here. You have agglutination, where you have a few different bacteria, and these antibodies are kind of forming cross-links with each other. And then precipitation is you have this long chain of antigens and antibodies, and this would ultimately settle out of solution, settle out of the blood, and form an insoluble precipitate. So we're going to stop here, and then we will pick up with cell-mediated immunity in the next video.